Okay, in the last video we went into some depth on the digestion and absorption and utilization of carbohydrates. So now let's dive into lipid digestion and absorption and utilization, which is going to be a little bit different from the other kinds of nutrients. It also begins sort of in the mouth. You do make an enzyme in your mouth besides salivary amylase. It's called lingual lipase. And it does break down triglycerides, which are what we call fats. And triglycerides aren't like carbohydrates or proteins or nucleic acids in that they don't have a monomer-polymer relationship where you have polymers that are made up of subunits of lots of individual monomers, right? Starch is made up of lots of glucose, as is glycogen. Your proteins are made up of lots of amino acids. Here we have triglycerides that are made of a glycerol and 3 fatty acids. And lingual lipase can begin that digestion in the mouth, but it doesn't really get started until the stomach, because lingual lipase operates optimally at a lower pH. And, our mouth is a relatively neutral pH. The stomach is a pretty low pH. So, in the stomach, chyme really mixes everything up with the hydrochloric acid and then the lingual lipase. And we break down some fats there. But despite the lingual lipase in the stomach, only about 20% of triglycerides are broken down by the time the chyme has left the stomach. That means 80% happens after the stomach, or more accurately, in the small intestines. And we'll talk about why that is in a moment. Once you get in the duodenum, as we talked about before, you release the hormone CCK from your duodenum, cholecystokinin, and cysto, right? That refers to the gallbladder. It's going to cause your gallbladder to squeeze and eject all of that stored bile into your duodenum. And bile is the big difference here. It's why only 20% of your fats are digested in your stomach and 80% are digested after. Fats are hydrophobic. They don't dissolve well in water, and so they tend to, even if you mix them up a lot like you do in the stomach, they will continue to separate, and you get these big globs of fats instead of lots of little pieces. Well, you can only digest, if you're lipase, the enzyme that breaks down lipids, around the surface, right? You can't, beam yourself into the middle of that glob and start digesting from the middle on out. You have to digest from the surface. And big globs have a lot less surface area than lots of smaller globs. Well, bile emulsifies those fats. And emulsification is sort of like what Dawn dish soap does to big globs of bacon grease in your pan, right? You have two ends of the molecule. It's amphipathic, meaning one end is hydrophobic, means it combined with the fats, but the other end is hydrophilic. It combined with the water, and those big globs get broken into much, much smaller globs, much more surface area for our pancreatic lipase to now break up the lipids. So, bile is essential for effective digestion of fats, but bile does not itself digest fats. I repeat, bile does not digest fats. What digests fats? Well, lipase does. In your small intestines, where most of this digestion occurs, it's pancreatic lipase. So at this point, the lipids that you have are surrounded by little, glycoproteins, bile salts, right, that surround the fats so that they can dissolve a little bit in the water, and these are called micelles, these big globs of smaller globs, I should say, of fat with bile salts around them. And those contact the intestinal epithelium, and those lipids, which are not triglycerides anymore, right? We broke them down. They are now a glycerol and 3 fatty acids. Those diffuse across the plasma membrane pretty easily because it's lipids. Those bile salts are then released, and we will return them eventually to the liver. But those triglycerides are then put back together in the cell. And I know that seems weird, right? We just broke them apart so that we could absorb them. Now we put them back together, and we package them with a bunch of new glycoproteins. And these proteins, lipoproteins, which are part lipid, part protein. These lipoproteins allow these lipids to be soluble in water for a time, right? Because the protein has a hydrophobic end that binds to the lipid and a hydrophilic end that can dissolve in water. So chylomicrons are these initial big old lipoproteins, and we pump them out into the tissue fluid inside that intestinal villus, so we've absorbed it across there, but it's too big to get picked up by the blood capillaries. And if the blood capillaries can't pick it up, how in the world is it going to get transported through the body? Well, it turns out that we pick them up, as we mentioned a few chapters ago, into lacteals. Right? Lacteals are lymphatic capillaries, which have these big flaps. They let in big old stuff, including bacteria and old cellular debris, but also these big chylomicrons. So they go through the lymphatic system first, actually, and they don't get dumped into the bloodstream until, your right lymphatic duct, and actually in this case it would be your thoracic duct, because we're coming from your intestinal, lymphatic capillaries, goes up and dumps into your subclavian vein, and now we are in your bloodstream. And actually then we're going to get processed by the liver, just as you might expect, just like most other things do. So most of these chylomicrons find their way to the liver, and the liver adjusts them and creates new lipoproteins. A couple of different kinds. One called low-density lipoproteins, and one called very low-density lipoproteins. And again, these lipoproteins are just proteins attached to lipids so that the lipids can dissolve in water. The low-density lipoproteins mean it's a lot more fat than protein, right? The very low-density lipoproteins mean it's a lot more lipid than protein. But the VLDLs really are transporting fats, and the LDLs are transporting cholesterol. So the VLDLs, very low-density lipoproteins, transport these fats, triglycerides, to muscles for use and adipose tissue for storage. The low-density lipoproteins deliver cholesterol, different kind of lipid, to just your peripheral tissues, your cells in your body. And those cells pick up that cholesterol and use it for all kinds of things. They use it to put in their cell membranes. They use it to make hormones and other materials. And if there's excess cholesterol, it just diffuses back into the bloodstream. Right? So when you get a cholesterol panel, you may have heard people talk about good cholesterol and bad cholesterol. These LDLs are usually what we talk about when we talk about bad cholesterol, because if you have too many of them, that excess cholesterol is going to get into the bloodstream, and if they're in the bloodstream, then they can end up getting into the walls, forming deposits in the walls of your blood vessels, and that's atherosclerosis, and it can cause all kinds of clotting and troubles. But you also make high-density lipoproteins, and high-density lipoproteins mean lots of proteins, very little actual cholesterol in there, very little actual lipid. And these high-density lipoproteins go out and they pick up excess cholesterol in the bloodstream that might have been dropped off by your LDLs, and they return it to your liver, where it is usually gotten rid of as bile salts, or we might reuse that cholesterol if we need it. So your ratio of HDLs to LDLs is really what matters when you have a cholesterol panel. You want more HDLs and you want fewer LDLs. And this is a nice summary figure in your book, which talks about what happens, right? We make VLDLs, which take triglycerides for storage in the adipose tissue, primarily. We have LDLs, which drop cholesterol at the tissues. Excess cholesterol goes to the bloodstream. We have HDLs that come pick up that excess cholesterol and take it back to the liver, where they came from, and dump it out as bile.